Massachusetts Technology Institute. Cancer cells can have thousands of mutations in their DNA. However, only a handful of those who really drive the progression of cancer. The rest is just for the trip. Distinguishing these mutations from the harmful driver of neutral passengers could help researchers identify better drug goals. To increase these efforts, a team led by the MIT has built a new computer model that can quickly scan the entire genome of cancer cells and identify mutations that occur more frequently than expected, suggesting that they are driving tumor growth. This type of prediction has been a challenge because some genomic regions have an extremely high frequency of passenger mutations, drowning the signal of real drivers. We created a probabilistic deep learning method that allowed us to obtain a really precise model of the number of passenger mutations that should exist anywhere in the genome, says Maxwell Sherman, a graduated student of the MIT. Then we can look throughout the genome in search of regions in which it has an unexpected accumulation of mutations, which suggests that these are drivers' mutations. In their new study, researchers found additional mutations throughout the genome that seem to contribute to tumor growth in 5 to 10% of cancer patients. The findings could help doctors identify medications that would have more chances of treating these patients successfully, researchers say. Currently, at least 30% of cancer patients do not have a detectable driving mutation that can be used to guide treatment. Sherman, the graduated student of MIT Adam Yori, and the former Mitiva Preeb research assistant are the main authors of the study, which appears today in the Biotechnology of Nature. Bonnie Berger, Simons mathematics professor at the MIT and director of the Computation and Biology Group in the Artificial Computer and Intelligence Laboratory, is the main author of the study, together with Poiu Loh, assistant professor at Harvard Medicine School of Medicine and associate member of the Broad Institute of MIT and Harvard. Felix Dietlein, an associated professor at Harvard's faculty and Boston Children's Hospital, is also the author of the newspaper. A new tool. Since the human genome sequenced 2 decades ago, researchers have been touring the genome to try to find mutations that contribute to cancer by making cells grow uncontrollably or evade the immune system. This has successfully produced objectives, such as the epidermal growth factor REC.